Hello, College Biology. This is Proteins Part 3, and we're going to discuss denaturing. So, let's talk about it right away. Denaturing is when a protein loses its functional shape and unravels, and that's usually not good for a cell. So, what would cause this to unravel? Well, a protein on its own, naturally, has these R groups. Some are attracted to each other, some repel, some are hydrophobic, some are hydrophilic, but on its own it will fold into its shape. So something from the outside has to affect it in order for it to unfold, because on its own it will fold. And so changes in environmental condition can influence the folding of a protein so that it loses its functional shape by unfolding. So what can cause that? Well, let's look at the folding. One of the primary components of protein folding is hydrogen bonds, these attractions of partially charged regions of one molecule and another molecule. So that's going to be about electromagnetism. When you have charges attracted to each other, that's because they've got polarity, they've got partial charges here, and they're attracted to each other. Well, all of these particles are moving. They all have a little bit of vibration. And we know that electromagnetism gets weaker at the greater distance you have, which is why you can have two magnets close together, and they'll snap together when you let go. But if you bring them farther apart and let go, well, they'll just drop. They're not going to be strong enough to pull each other. So the farther the distance, the weaker the attraction. Well, what happens is one of the environmental conditions that can affect denaturing is temperature. Temperature is a measurement of heat, and heat is about the motion of molecules. So we've got in this diagram, we've got some speed lines to indicate how fast we're going. We've got these short speed lines over here to show that these particles are slow. We've got these fast speed lines over here to show that these particles are zooming around. So what does that have to do with us? Well, if we go back here and we heat this up, we transfer heat to this molecule, we have more heat. Well, what's going to happen is the particles that are attracted to each other because they're really close are going to start to vibrate. They're going to start to vibrate. And when they vibrate, as they increase in heat, there are going to be times where they're farther apart from each other, and at some point, they're going to be far enough apart from each other that they are too far apart to feel that attraction anymore, that electromagnetic attraction. And so now they are not bound to each other. They're zooming around out here, far apart from each other, not attracted to each other. And that can contribute to unfolding, and when that happens, the protein loses its function, and it's not going to be able to do whatever the cell needs it to do. So regulating temperature is very important for the cell so that the proteins are in the right condition for them to work. That is one factor that contributes to denaturing. There is a second factor that contributes to denaturing, and that is ions in the environment. So if a protein is in water, that means that there are still some ions around, but there's not too many. The protein can deal with them, and they can fold into their natural shape. However, if we have a bunch of other ions surrounding that protein, just floating around near the protein like we have over here, just ions floating around, well, the electromagnetic attraction of those ions is just as strong, if not stronger, than the electromagnetic attraction of the R groups, which means that there might be an R group over here that's kind of, right now, it has a hydrogen bond to the second R group, but when this hydrogen comes along, it says, oh no, we're attracted to that. And so, bam, you've got your hydrogen ions, in this case hydrogen ions, attracting to those R groups. So if you have a lot of ions in the environment, they will compete with the R groups for attraction. And if the R groups are attracted to the ions instead of each other, then they're not going to fold into their shape. In fact, they'll lose their shape. They will unfold, they will denature, and the protein will not be able to do its job. That's exactly what we're seeing here. We have ions in the environment that get attracted to specific R groups, and then those R groups aren't going to bind with each other, and the protein doesn't have its shape anymore. So what is going to increase ions in the environment? Two things, really. One, pH. pH, acidity, basicness of the environment. If you have a very low pH, that means you have a lot of hydrogen ions in that environment. If you have a very high pH, that means you have probably a lot of hydroxide ions in that environment. Hydroxide is negatively charged, and it doesn't really matter if these ions are positive or negatively charged. Both will compete for polar regions, and both will involve hydrophobic interactions with non-polar regions. So it doesn't matter if they're positive or negative. A third thing, and so that's acidity. If you've got a highly acidic, low pH, you got hydrogen ions. If you've got basic, high pH, you've got hydroxide ions. The last thing is salt concentrations. If you add a bunch of salt, we can use table salt here, sodium chloride. When they get in that water, they'll dissociate into salt, obviously sodium positive ion and chlorine negative ion. And now you've got a bunch of ions in there, and it doesn't matter if they're positive or negative. They'll both compete with the R groups, causing the protein to unravel, losing the protein's shape. We'll talk about renaturation in our next video. I hope this helps. Struggle well.